You know, that feeling when you're just firing on all cylinders, feeling really vibrant. Or maybe you've faced a health issue and wondered, what's actually going on inside me?
Hmm.
It's kind of mind-blowing when you think about the incredibly complex molecular dance happening inside us every single second. This hidden world, it dictates everything, right?
Mm.
Our energy, how we
Yeah.
fight illness.
And that really is biochemistry. We've got some foundational texts here, the original work that, laid the groundwork for how we understand life at its most basic level
Yeah.
and how all those molecular bits relate to health and disease.
Right.
Our goal isn't just a quick skim. We're aiming for a deep, sort of intuitive grasp of how biochemistry and medicine are, well, completely tangled up together.
Exactly. We're not just trying to memorize a list of molecules here. We want to get the story, the epic story of discovery, the key moments that really set up biochemistry as its own powerful science. And then, crucially, map out how this, constantly changing biochemical knowledge keeps reshaping how we understand and treat the human body. So, yeah, get ready to journey with us right down to the molecular level. This is where life really happens.
Okay, so our exploration kicks off in the late 19th century. It's a time when even something that seems pretty straightforward now, like fermentation, was still a deep mystery.
Yeah, it's amazing to think people watched sugar turn into alcohol for centuries without really knowing how.
Absolutely. And the main idea, pushed heavily by, the big name Louis Pasteur, was that this process needed living yeast cells, intact cells.
Right, like there was some kind of vital spark, some magic life force required.
Exactly. But that whole idea got turned on its head in 1899. Thanks to the, well, really groundbreaking work of the Buchner brothers, Edward and Hahn.
Ah, the Bootners.
Yeah. In a really pivotal discovery, they showed that a cell-free extract of yeast could still ferment sugar. The story goes they were trying to preserve this yeast extract using lots of sugar. And kind of unexpectedly, the mixture started fermenting. Even though the yeast cells were deliberately broken open, apparently it bubbled up and spilled all over their lab bench. A bit of a messy accident, but with huge consequences.
Huh, a lucky spill then. But that seemingly random observation, it had a massive impact, didn't it? It completely flipped the script.
Oh, completely. It was a watershed moment. This discovery is really seen as the birth of biochemistry as its own science. It proved definitively that biological processes, even something complex like fermentation, could be studied outside living cells,
Wow.
just through the actions of the molecules themselves. It opened up this entirely new and incredibly productive way to investigate the basic chemistry of life. It shattered that old vitalistic view, showed that life's processes follow chemical rules. That's fundamental.
So once they knew these cell-free extracts were the key, it sounds like things really took off. A cascade of discoveries.
Totally. The years right after the Buchner's work were just packed with crucial findings. Researchers quickly started pinning down the specific molecules involved in fermentation and broader cell metabolism.
Like what?
Well, think about inorganic phosphate, ADP, and ATP, the energy currency, basically, that runs everything in the cell.
Right. ATP. You hear about that all the time.
Exactly. And the roles of NAD and NADH, key players in electron transfer, the basis of how cells get energy from food, those were figured out too. Plus, all sorts of phosphorylated sugar intermediates the steps in breaking down glucose.
Yeah.
These early finds revealed the basic building blocks and energy carriers, like that ATP that power, well, pretty much all life.
These are molecules we see in Bio 101 now, but back then they were totally new concepts, weren't they? Realizing specific molecules did the work must have been revolutionary.
Absolutely. And this first wave gave them the foundation to understand the bigger, more complex metabolic pathways. The whole step-by-step breakdown of glucose glycolysis that was mapped out. And the citric acid cycle, the sort of central hub where fuel gets completely oxidized for energy. Even things that seem a bit separate, like how our bodies get rid of nitrogen waste through urea biosynthesis that was unveiled at the molecular level too. It's pretty incredible, piecing together these complex, interconnected chemical roadmaps, step by painstaking step.
It really shows the power of that reductionist approach in biology, right? Breaking things down to understand the whole.
Definitely. And it didn't stop there. The next few decades saw the discovery of essential vitamin-derived cofactors and coenzymes.
Cofactors.
Coenzymes. Yeah, these are small organic molecules that basically help enzymes do their jobs. They bind to the enzyme and assist in the chemical reaction. We learned about thiamine pyrophosphate crucial for carb metabolism.
Hmm.
Riboflavin, which leads to FAD and FMN, vital for electron transfer. Coenzyme A, central to fatty acid breakdown and the citric acid cycle. Coenzyme Q, or ubiquinone, a key electron carrier for making ATP. And the interesting cobamide coenzyme from vitamin B12 involved in specific rearrangements and methyl transfers.
So an enzyme is like the tool.
Exactly. And the coenzyme is like the essential battery or bit that makes the tool work.
So vitamins weren't just some vague good things in our diet. They were being revealed as the direct precursors for these molecular helpers doing critical jobs.
Precisely. Then the 1950s brought more big breakthroughs. Understanding how our bodies build up and break down complex carbs, assembling them, taking them apart for energy.
Okay.
The pathways for making pentoses, those 5 carbon sugars needed for DNA and RNA, were worked out. And at the same time, the complex ways amino acids and fatty acids are broken down, catabolized for energy or other building blocks, were mapped in detail. It was this period of intense map-making of the cell's molecular landscape.
And these discoveries weren't just happening by observation anymore. You needed new, more sophisticated tools and techniques to make these jumps, right?
Oh, absolutely crucial. Researchers started using a whole range of powerful experimental systems. Animal models let them study pathways in a whole organism. Perfused organs running fluid through an isolated organ allowed study of organ-specific functions in a controlled way. Tissue slices were simpler systems. And cell homogenates, basically just busting cells open, and their subfractions, like separating out the mitochondria or ribosomes, let them isolate and study specific parts and their biochemical jobs. And of course, purifying individual enzymes was key to understanding exactly what each one did.
So they were systematically taking the cell apart, literally and figuratively, to figure out how it worked molecule by molecule.
Exactly. And alongside these lab techniques came vital tech innovations for analyzing and separating molecules. Analytical ultracentrifugation, spinning things incredibly fast, separated molecules by size and shape, and various types of chromatography paper, ion exchange, gel filtration. These were powerful tools for sorting out complex mixtures of molecules based on their different properties, like sorting different colored or sized beads.
And then the atomic age arrives, and that brought a whole new toolkit for actually tracking molecules inside systems.
Yes, the post-WWII era saw the widespread use of radioisotopes. Carbon-14, tritium, which is hydrogen-3, phosphorus-32. These radioactive tracers were invaluable.
How so?
Well, you could track the precise movement of atoms and molecules through complex pathways. A classic example is figuring out cholesterol biosynthesis. Researchers could feed cells labeled precursors and then literally follow where those radioactive atoms ended up in the final cholesterol molecule. It revealed the exact sequence of steps.
It's like following, glowing breadcrumbs through the metabolic maze.
That's a great way to put it. Exactly. And then, to really see the 3D shape of the molecules, you needed X-ray crystallography.
Right, to see the structure.
By shooting X-rays through crystallized biomolecules and analyzing the patterns, scientists could figure out the precise 3D structures of proteins, DNA, RNA, enzymes, even viruses. Seeing the structure gave critical clues about how they actually worked.
Seeing the lock and key of an enzyme, for example.
Absolutely transformative. And then, building on all this structural and functional knowledge, came the genetic revolution after the DNA double helix discovery. That led to tools like PCR, the polymerase chain reaction.
Which lets you amplify DNA sequences, right?
Yeah.
You have lots of copies.
Millions of copies from a tiny sample. It also enabled creating transgenic animals with foreign DNA added and gene knockout animals where specific genes are switched off. These tools let scientists directly study what individual genes and their protein products do.
So not just seeing the machinery, but being able to tweak the blueprints.
Precisely. And this toolkit just keeps getting better. We're constantly developing new ways to prepare, analyze, purify, identify all the metabolites in a cell, the small molecules involved in metabolism, and refining techniques to study the activities and 3D structures of natural enzymes, and also recombinant enzymes, ones made in the lab using genetic engineering. It's this continuous cycle. Tech innovation drives deeper understanding.
Okay, so we've traced how biochemistry emerged as this distinct, powerful science driven by these amazing discoveries and tech leaps. Now let's shift to our second big theme, the deep, inseparable link between biochemistry and medicine. You called it a two-way street. Let's get into some examples to really appreciate that interplay.
Indeed, it's not just biochem providing info for medicine, it's dynamic, reciprocal. Understanding biochemical principles, it deeply informs how we see health and disease. But just as importantly, studying diseases often uncovers totally new areas of basic biochemical research.
You mentioned some really good early examples of this. Let's revisit those. Sickle cell anemia is a classic, isn't it?
It really is a landmark.
Yeah.
Discovering the difference between normal and sickle cell hemoglobin was just a single amino acid change in the protein chain that was huge.
Just one change.
Just one. But it didn't stop there. Studying lots of other natural hemoglobin variants, each with its own slight change, really boosted our understanding of how protein structure dictates function. It showed so elegantly how tiny molecular changes can have massive, sometimes devastating effects on the body.
And then there was Archibald Garrod's work on inborn errors of metabolism. Sounds like that really laid the groundwork for understanding the genetic basis of many diseases.
Truly foundational. Early 1900s, Dr. Garrod studied patients with rare conditions like alkaptonuria, albinism, cystinuria, pentasuria.
Okay.
Through sharp clinical observation and clever chemical analysis of their body fluids, he showed these were genetic caused by specific inherited defects in certain biochemical pathways. He coined that phrase, inborn errors of metabolism. His work basically launched the field of human biochemical genetics.
Wow.
It was the dawn of realizing many diseases start with specific molecular flaws passed down through families. So studying these rare conditions gave insights into fundamental metabolic processes everyone needs.
So by looking closely at uncommon diseases, Garrod unlocked basic insights relevant to everyone's health. That's a powerful example of disease study illuminating basic biology.
Exactly. A more recent example that shows this two-way street beautifully is familial hypercholesterolemia.
High cholesterol that runs in families.
Right, a common genetic disorder causing very high cholesterol leading to early heart disease. Biochemical research didn't just pinpoint the gene mutations defects in the LDL receptor gene, it also gave us a much deeper understanding of cell surface receptors in general.
How so?
Specifically the LDL receptor, yes, but also the whole mechanism of receptor-mediated endocytosis, how cells bring specific things in from outside. Not just cholesterol, but lots of essential stuff. So studying a cholesterol disease led to fundamental breakthroughs in how cells internalize all sorts of molecules.
That's fascinating. A cholesterol problem leads to understanding how cells eat at a molecular level. You also mentioned oncogenes and tumor suppressor genes in cancer. How does that fit?
Oh, the research into oncogenes and tumor suppressors in cancer cells has been absolutely key to figuring out the complex molecular controls for normal cell growth and division. It showed how these pathways go wrong in cancer, leading to that uncontrolled growth. Understanding these biochemical pathways and the specific molecular changes in cancer is crucial for designing targeted therapies that actually attack the malfunctioning bits.
Right, hitting the specific problem.
These examples really drive home how looking at disease clinically and cellularly can spark fundamental, often unexpected biochemical discoveries. It just underlines that a solid science foundation is the bedrock for smart medical practice. It's not just treating symptoms, it's understanding the why at the molecular level.
It highlights how scientific thinking fuels curiosity in healthcare pros, fosters that continuous learning mindset, right? Now, let's pivot slightly. How does biochemistry impact something so everyday like nutrition and the rise of preventative medicine?
Well, biochemically, you can think of health as this dynamic balance, right? Where all the countless biochemical reactions inside and outside cells are running optimally, keeping things stable, letting the organism thrive.
Homeostasis.
Exactly. And maintaining that health needs the right dietary intake of essential nutrients, vitamins, certain amino acids and fatty acids we can't make ourselves, minerals, water. Our whole understanding of nutrition is completely rooted in biochemistry. The two fields share that focus on the essential chemicals for life. So when we talk about a balanced diet, we're really talking about giving our bodies the specific biochemical building blocks and cofactors needed for everything to work right at the molecular level.
So needing vitamins and minerals isn't just vague, good-for-you advice. It's about providing specific molecules for specific chemical jobs in the body.
Precisely. And there's this growing, really important emphasis now on preventative medicine, using nutrition and lifestyle changes to stop chronic diseases like atherosclerosis, cancers, type 2 diabetes before they start.
Being proactive.
Right.
This focus comes from our better understanding of the specific biochemical roles different dietary bits play in either causing or preventing these complex diseases. It's a big shift from just treating illness to actively maintaining health at that fundamental molecular level.
A real paradigm shift from reaction to proaction, all based on biochemical understanding. Now, you said most diseases, apart from infections or pollutants, have a biochemical basis. Can you break down the different ways these biochemical problems show up?
Sure. Think about something like an electrolyte imbalance. That's a disruption in the biochemical balance of ions like sodium, potassium, calcium, absolutely vital for nerves and muscles.
Okay.
Or defective nutrient intake or absorption, maybe diet's poor, or the gut isn't absorbing properly. That directly limits the biochemical building blocks and helpers the body needs.
Makes sense.
Hormonal imbalances, too much or too little of a hormone, can throw whole biochemical pathways out of whack. Toxic agents, chemical or biological, can directly mess with reactions, maybe inhibit an enzyme or damage molecules.
And of course, genetic disorders, DNA-based problems lead to faulty or missing proteins or enzymes. That disrupts entire pathways, causing all sorts of diseases. It just shows how interconnected everything is. A small glitch somewhere can ripple right through the system.
It really underscores the complexity, the delicate balance needed to sustain life. One disruption can have widespread effects.
Totally. And tackling these complex biochemical challenges needs an increasingly interdisciplinary approach now. Modern biochem research is deeply tied up with genetics, cell biology, immunology, nutrition, pathology, pharmacology. You need insights from all these fields.
It's a team effort, different scientific expertise coming together.
Exactly. And it's worth remembering, biochemists aren't only focused on human health. They're also involved in tackling bigger global issues, finding sustainable solutions for humanity, environmental challenges, and also, importantly, helping educate the public about the scientific method itself. That's vital for good decision-making everywhere.
That brings us to a truly massive milestone in both biochemistry and medicine, the Human Genome Project. This feels like a real turning point, a game changer.
Oh, an absolute paradigm shift. The progress in sequencing the entire human genome in the late 90s was remarkably fast. It led to the announcement in the early 2000s that, over 90% was done.
Wow.
This huge international effort was mainly the publicly funded International Human Genome Sequencing Consortium and also the private company Cellular Genomics. The finished sequence was declared in 2003, just 50 years after Watson and Crick described the double helix.
50 years, that's pretty quick in scientific terms for something so massive.
Yeah.
And you said the implications were virtually unlimited. Can you unpack the potential that unlocked?
Right. The ability to isolate and sequence any gene in the human genome and then use that info to study its structure, function, regulation, maybe using gene knockout experiments. It just opened up previously unimaginable research avenues.
Yeah.
Scientists could find totally new genes and their proteins, leading to huge insights into development, physiology, evolution. And crucially, it gave us powerful tools to find the specific genes linked to susceptibility or development of countless human diseases.
So it wasn't just having the static sequence, the letters.
No. It was the dynamic ability to read it, understand it, manipulate it, study the genes and what they do.
Got it. Not just the blueprint, but the ability to read and edit it.
Exactly. And the progress keeps rolling through studying genome mutations in various model organisms.
Like fruit flies and worms.
Yeah, yeast, Saccharomyces cerevisiae, the fruit fly, Drosophila, the worm, C. elegans, zebrafish. These are often simpler genetically, easier to manipulate in the lab. They're powerful systems for understanding basic gene functions and mechanisms. And often, discoveries in these models give us really valuable clues for understanding and maybe treating complex human diseases like cancer or Alzheimer's. They're like simplified but informative test systems relevant to us.
Like having a Rosetta Stone for basic genetic principles across species.
That's a good way to think about it. And the Human Genome Project directly spawned and massively accelerated what we now call the IMIX fields.
Ah, yes, IMIX.
These are cutting-edge areas focused on the comprehensive, systematic study of entire sets of molecules in a biological system, structures, functions, interactions. It's a fundamental shift from looking at one gene or protein at a time to analyzing whole networks.
The IMIX revolution sounds like a whole new biology language. Can you break down the main ones? What does each arm focus on?
Sure. Transcriptomics looks at the transcriptome, all the RNA molecules, messenger RNA, transfer RNA, ribosomal, non-coding RNAs, produced by the genome at a specific time under specific conditions.
Go with the RNA picture.
Proteomics, as the name suggests, deals with the proteome, the entire set of proteins, including modifications and interactions, in a cell or organism at a given time.
The protein picture.
Glycomics studies the glycome, all the carbohydrates, simple and complex, their structure, how they're made, their roles. Lipidomics focuses on the lipidome, the complete set of lipids, those fatty molecules.
Carbs and fat.
Metabolomics investigates the metabolome, all the small molecule metabolites like sugars, amino acids, lipids involved in metabolism. Nutrigenomics explores how nutrients affect gene expression and how our individual genetic variations affect how we process nutrients.
Nutrition and genes interacting.
And pharmacogenomics uses genomic info to optimize drug discovery and development, aiming to tailor treatments to your specific genetic makeup for better results and fewer side effects.
Personalized medicine, essentially.
Exactly. While transcriptomics tells us which genes are on by looking at RNA, proteomics shows us the actual protein machinery doing the work.
That's an incredibly detailed toolkit for understanding life's complexity from almost every angle. You mentioned a striking example from transcriptomics.
Yes, the really spectacular explosion of knowledge about small, non-coding RNA molecules. These tiny RNAs, often overlooked before, turn out to be critical regulators of gene activity.
How so?
They play incredibly diverse roles in controlling which genes get turned on or off, when, how much. It adds this whole other intricate layer to how gene expression is controlled, like finding a new set of hidden switches and dimmer knobs for our genes.
Like a hidden regulatory language within the cell using these small RNAs.
Exactly. And handling, analyzing, making sense of the absolutely massive amounts of data pouring out of all these RMX fields needs a powerful partner. Bioinformatics.
Right, the computational side.
Bioinformatics develops the computational tools and algorithms to collect, store, organize, analyze, and interpret all this biological data, DNA, RNA, protein sequences, metabolic data. Without bioinformatics, we just drown in information from these high-throughput technologies. It's the essential data science underpinning all this.
So bioinformatics is the critical infrastructure making sense of the omics data flood.
Precisely. And the momentum from the Human Genome Project also boosted several related fields. Biotechnology uses biochemical, molecular, genetic engineering approaches for products in medicine, agriculture, industry. Bioengineering applies engineering principles to biology and medicine, developing new tech devices. Biophysics uses physics principles and techniques to study biological systems, from molecules up to organisms.
And bioethics.
Crucially, bioethics addresses the ethical implications and societal impact of all these powerful new biological and medical technologies. We absolutely need that ethical framework.
Essential to consider as capabilities grow so fast. Now, you mentioned specific applications that came from the HGP and omics. Molecular diagnostics. How has that changed things?
Molecular diagnostics uses techniques like DNA probes, PCR assays to help diagnose and monitor a huge range of conditions: biochemical disorders, genetic diseases, infections, immune disorders. Analyzing DNA, RNA, proteins quickly and accurately has revolutionized diagnosis, risk assessment, predicting treatment response, monitoring therapy.
And nanotechnology, manipulating things at the nanoscale. How's that used in medicine?
Nanotechnology works with structures on the scale of 1 to 100 nanometers. In medicine, this means things like nanoparticles or nanoshells for highly targeted drug delivery right to diseased cells, or for new diagnostic tools, maybe in cancer therapy.
Tiny machines for precise medical jobs. What about stem cell biology? Lots of buzz around that.
Stem cells are these unique, undifferentiated cells. They can renew themselves, make more copies, and differentiate into any specialized cell type in the body.
Right.
Stem cell biology studies these cells and their huge potential for regenerative medicine, repairing damaged tissues, maybe even growing new organs eventually.
The potential to fix damage or grow replacements sounds incredible. And gene therapy, treating the root cause of genetic problems.
Gene therapy aims to treat or cure diseases by putting engineered genes into a patient's cells. The potential is enormous, especially for inherited disorders. But honestly, translating that promise into widespread clinical reality has been harder than first thought.
Still challenges there.
Yeah, but research is making progress. Better ways to deliver genes, better gene editing tech are being developed. It's still a very active area.
Still lots of R&D, but maybe transformative treatments down the line. Synthetic biology was another term you mentioned.
Synthetic biology combines molecular biology, genetic engineering, and engineering principles to design and build new biological functions or systems that don't exist naturally.
Like building biological machines.
Kind of. A cool example is engineering microbes to do useful tasks, like cleaning up oil spills, bioremediation. It's about designing and constructing new biological parts and systems.
Applying engineering to biology design. And finally, systems biology sounds like it brings everything together.
Systems biology takes a holistic view. It looks at complex biological systems as integrated, interacting networks, genes, proteins, metabolites, rather than just isolated parts.
Seeing the whole picture.
Yeah, understanding how all these players interact to create the properties and behaviors of the whole living system. Moving beyond parts to understand the functional whole.
Okay. You also mentioned an appendix listing important biomedical databases. Can you give us a quick tour of some key ones?
Absolutely. These public databases are critical resources for researchers everywhere. ENCODE, the Encyclopedia of DNA Elements, aims to identify all functional parts of the human genome.
Oh, yeah.
ISDB is the International Sequence Database Collaboration, linking major DNA databases from Japan, Europe, and GenBank in the US. PDB, the Protein Data Bank, stores 3D structures for countless proteins, DNA, RNA, etc.
The structural library.
GenBank from the NIH here in the US holds all publicly available DNA sequences and their protein translations. And HapMap, the International HapMap Project, mapped common patterns of human genetic variation, SNPs linked to disease susceptibility and drug responses. These databases are like massive, ever-growing libraries of molecular information used by scientists worldwide daily.
Invaluable repositories of molecular knowledge, constantly updated and accessed globally.
Precisely. All these advances, ticked off by the Human Genome Project and fueled by the intertwined revolution, are just undeniably making the 21st century an incredibly exciting, transformative time for biology and medicine.
This has been an absolutely incredible, really comprehensive journey through biochemistry's foundations and its deep, inseparable connection to medicine. To really cement our understanding, can we maybe summarize the key principles and relationships we've covered?
Definitely. So, at its heart, biochemistry is the science of molecules and living things, their reactions, the enzymes that catalyze them, how metabolic processes are regulated. It really is the basic language of all biological sciences. And while we focused a lot on humans because of the medicine link, it's vital to remember these principles apply across all life. From viruses and bacteria right up to complex organisms like us, the basic rules are universal.
So the fundamental biochemical playbook is the same across all life forms.
Absolutely, and that intimate two-way relationship between biochem, medicine, and other health fields, you just can't overstate it. Health, fundamentally, depends on all those biochemical reactions running smoothly, in balance.
Right.
And disease almost always reflects some kind of abnormality in molecules, reactions, processes. It's that incredibly delicate molecular balance that keeps us healthy.
That intricate molecular dance keeps us well, and missteps lead to illness.
Exactly. Advances in biochem knowledge have consistently lit up mysteries about physiology and disease causes. And conversely, like we saw, studying specific diseases has often uncovered totally new areas of basic biochemistry. Biochemical approaches are fundamental for understanding disease causes and designing effective therapies. Plus, biochemical lab tests are indispensable for diagnosis and monitoring treatment. So a strong foundation in biochemistry, along with related sciences, is just essential for rational, effective medical practice. And finally, the ongoing impact of the Human Genome Project and all the research it sparked will keep profoundly shaping biology, medicine, and health sciences for, well, decades to come. With that research in model organisms continuing to give us crucial clues for human diseases.
This deep dive has truly illuminated that fundamental molecular basis of life and health. We've traced biochemistry's fascinating history and really hammered home its inseparable evolving link with how we understand and treat disease.
And now, thinking about the incredible, relentless pace of discovery in the racemics and related fields we talked about, it really begs a big question for you, the listener. Given all this progress, what do you see as the most exciting, maybe potentially transformative future possibilities emerging at that intersection of biochemistry and medicine in the coming years?
A really powerful thought to mull over as we wrap up. For those of you whose curiosity has been sparked, whose understanding deepened by this dive, I really encourage you, dig deeper into areas that grabbed you.
Yeah.
Maybe explore some of those metabolic pathways we mentioned, research the omics fields more, or even check out some of those biomedical databases we listed. The world of biochemistry is vast and always expanding, offering endless chances to learn more.
Indeed. The ongoing evolution of biochemical knowledge is continuous, it's dynamic, and its profound impact on our understanding of life and its power to shape the future of health and medicine, well, that's only going to keep growing.